Hello, everyone. Thanks for coming to my talk. My name is Andrew Ferns. I am a second-year PhD student in biomedical science in the Department of Pharmacology and Neuroscience at the University of North Texas Health Science Center in Fort Worth. So I started my scientific journey as a synthetic chemist in Oregon. I graduated with a bachelor's of science in chemistry from Western Oregon University. And as a chemist, I've always had a fascination with compounds that could profoundly alter consciousness and cognition. And so that fascination led me to pursue a PhD in neuroscience with the goal of building a career in psychedelic research. However, I was accepted to a university in Texas with no history of psychedelic research, and I joined a lab in a field that on its face has almost nothing to do with psychedelics. So our lab is primarily focused on biogerontology, which is the scientific study of the biological mechanisms that underlie aging. And more importantly, it's the search for interventions that slow, prevent, or reverse age-related dysfunction. So in plain terms, we want to know why brains and bodies break down as they get older and what we can actually do about it. So we work mostly with aged mice because they let us ask questions on a time scale that we can't do in humans. And because the things that can go wrong in an old mouse brain will often look a lot like the things that can go wrong in the human brain. So this particular line of work is incredibly relevant today because we are currently experiencing a global demographic shift towards an aging population. So you can see from this data from the US Census Bureau that the number of people over the age of 65 has been rapidly increasing. At the last census in 2020, 1 in 6 people in the US was over the age of 60. So this amounts to about 1.1 billion people worldwide, and this number is expected to double in the next 20 to 25 years. In 2021, there were 57 million people worldwide living with dementia, and this carries with it a significant socioeconomic burden with the global cost of treating older adults at over $1 trillion. Caregiver burden is high as well, with these people being at higher risk for social isolation, financial burden, and poor health. And so what this really means in the big picture is that more and more people will be at risk for age-related cognitive dysfunction, and there are currently no reliable interventions that slow or delay this decline. So my interest in consciousness and cognition naturally led me to want to study cognitive decline, and my lab turned out to be the perfect place to ask questions that very few people in the industry were asking, which is what could psychedelics do for the aging brain? When most people think of cognitive decline and aging, they think of the memory loss that commonly occurs in Alzheimer's or other neurodegenerative disease, but it's often more complex than that. So as we age, we can begin to experience deficits in several areas of cognition, including learning and memory, attention, and executive function, which would include things like decision-making or being able to handle a monthly budget. Older adults can also experience declines in language processing. So this is things like remembering what items around your house might be called. And they can also experience changes to social cognition. So for example, not being able to interpret social cues or others' emotions. There's many factors that contribute to this decline, and we still don't know exactly what those causes are. But one thing that seems to be a major contributor is changes to the balance of excitation and inhibition in the brain. So if you think of your car, it has a gas pedal that makes the car go and a brake pedal that makes the car stop. Your brain works in a similar way. It has special cells called neurons that send go signals, which we call excitatory, and it has neurons that send stop signals, which we call inhibitory. And so just like your brake pedal can prevent you from driving off a cliff, your inhibitory neurons prevent your brain from receiving too much excitation. Just like everything in the body, though, the brain does rely on precise balance between the excitatory signals and the inhibitory signals. So if we think back to that car analogy, as your car gets older, the brake pads begin to wear down and your brakes don't have that same stopping power that they did when they were new. And so as we get older, these inhibitory neurons begin to lose function as well, and they're not able to stop the excitatory neurons as efficiently. So as we age, we begin to see an increase in excitation and a decrease in inhibition. This balance is how neurons communicate with each other, and if that balance is thrown off, those connections between neurons become less efficient at communicating with one another. And so you can think of this like a bad connection on a phone call where you're only catching every other word that the person's saying. If the neurons can't communicate with each other properly, then cognitive function suffers. And so I wanted to know if psychedelics could help repair the brain's braking system. I'm sure you're all aware that one of the major findings in psychedelic research is that their ability to increase neuroplasticity, which is the brain's ability to form new connections between neurons. We've seen this across several studies. You may even recognize some of these papers. And so I thought maybe we could harness this ability to try to reform some of the connections that are lost due to aging. And so there is a newer theory that's been proposed recently that psychedelics may be able to accomplish this through a two-phase mechanism. Psychedelics are typically thought to increase activity in those excitatory neurons that I mentioned before. But since the brain is always trying to maintain balance, myself and others think that this initial increase in excitation could cause something like a rebound effect, where the inhibitory neurons try to compensate for the extra excitatory signals by increasing their inhibition in an effort to try to keep things in balance. And that with the help of the neuroplasticity effects of psychedelics, this could form new connections in the brain that ultimately help repair that braking system permanently. And as I explained, this rebalancing of the excitation and inhibition balance in the brain could help improve the way neurons communicate, which could ultimately lead to improved cognitive function. So we're using the compound 1P-LSD to test this as an intervention for age-related cognitive decline. 1P-LSD is the chemical cousin of LSD, which I'm sure we're all familiar with. It just has a few extra atoms attached, and by itself, it's not very potent. But fortunately, once ingested, the body converts this drug to LSD. Another benefit of using this compound is that it's currently unscheduled in the US at the federal level. It does fall under the Federal Analog Act, but can still be purchased and used for non-human research, which allows us to effectively study LSD without the regulatory hurdles associated with Schedule I compounds. Another question I had is, does the therapeutic benefit of psychedelics require the subjective experience? So what I mean here by subjective experience is the sensory perceptions, emotional changes, and shifts in self-awareness that emerge with psychedelic use at higher doses. And it's these subjective effects that are often utilized during psychedelic-assisted psychotherapies to bring about the changes in thinking that lead to these positive clinical outcomes. But these higher doses may not be necessary to induce the beneficial cellular and molecular effects. So our lab is interested in whether or not these low doses can still induce plasticity and alterations to brain activity. So we're using a low dosing regimen. This is something akin to microdosing, where we're giving aged mice a low dose of the drug every 4 days, and we did this for 90 days total. During that time, they underwent several different tests that are designed to measure their cognitive and motor functions. So this is what our experimental timeline looks like over the 90-day dosing and testing period. I know it looks a little complicated, but this is really just to illustrate the amount of work that went into this project. So we had 19 total male and 17 total females in this pilot study, and they were all aged 18 months at the start of testing. So it's important to keep in mind mice don't age at the same rate that humans do, but 18 months in mice is about equivalent to age 55 to 65 in humans. And we use this age because this is the average age in humans where cognitive decline first becomes noticeable in the clinic, and this is when any treatment would then likely be started. So like I mentioned, the animals underwent a large battery of both cognitive behavioral tests and tests of motor function. And they were given injections of either our vehicle, which contained no drug, or 0.1 milligram per kilogram 1P-LSD every 4 days for 90 days. And we use this dosing schedule to avoid any tolerance from the drug that's commonly associated with repeated dosing of psychedelics. So I did want to keep this presentation pretty easy to understand, so I'm not going to show a whole lot of data, but I did want to show some promising preliminary results that we have. So in a second, I will show you results from our T-Maze tests. So this is a discriminated avoidance task, which means that this test measures how well the animal can learn to avoid something unpleasant. So this is similar to how you might learn to avoid a certain route from home because the traffic is always bad on that route. So the mouse is placed into this T-shaped maze and has to learn to navigate to one arm of the maze in under 5 seconds to avoid a mild foot shock. And so let's call that the arm with the green check. That's the correct arm. So this initial learning phase is called the acquisition phase. Once they've learned to do that, we do what's called the reversal phase, which is where we switch which arm is the correct or safe arm, and we retest them to test their cognitive flexibility, which is their ability to change their behavior in different circumstances. So you can think of this like if there's suddenly construction on your route home from work, then you need to decide to go a different way. So here is the data. And in these graphs, a lower number or a smaller bar is going to indicate better performance or improvements on the task. So on the y-axis, we have trials to criterion, and we measure this as the number of trials it takes the mouse to choose the correct arm 4 out of 5 times in a row. And then we have our treatment groups on the x-axis. And so we are beginning to see an effect of the treatment where the mice given 1-PLSD are learning the task faster than the mice who did not receive the drug. So that's this top graph, which is the acquisition phase. We also see an effect on the reversal phase, which is the bottom graph, where the treatment group is able to learn that the safe arm is no longer safe, and they're learning this faster than the mice that did not receive the drug. And this is where we're measuring that cognitive flexibility, or that ability to change their behavior under new rules or circumstances. So just as a summary of what we've observed in our study so far, we do see improvements to learning and cognitive flexibility that I just showed you. And interestingly, it did seem like the effect was consistently greater in the female group, which could suggest a possible sex-dependent sensitivity to the drug. This could be due to different receptor expression levels in males versus females or hormonal fluctuations. We don't really know yet, but we're planning to look into this more into the future. And I didn't have time to go over the motor function tests or any of those results, but we did also see some improvements to fine motor control and balance. And if you're interested in learning more about that, you can be sure to contact me to discuss. So my next step for this project over the course of my PhD work will be to figure out the why. So how are these compounds working at the cellular level to produce those effects that we are seeing at the behavioral level? And do these effects indeed alter the excitation and inhibition balance in the brain, leading to improved cognitive function as we have hypothesized? So I just wanted to close this by asking you to think back to those numbers that I gave you at the start. With the amount of people affected by cognitive decline and dementia, it's likely that someone you know will experience this. And for those of you who already know someone suffering from dementia, you know how hard it can be on the patient and their family. So my work is really just about the possibility of giving the people we love more good, healthy years. And with how fast our population is aging, it's important that we continue this type of research and find something that will work. I don't want to promote this as a miracle cure because this work is preliminary and it is in mice. But to me, it is very exciting, and I'm looking forward to getting more data to share with everyone in the future, and I hope you will stay tuned for that. So with that, I would like to acknowledge my major professor, Dr. Natalie Sumian, my past and current lab members, my graduate advisory committee. These people have all been so great to me, and I'm thankful for the freedom that they've given me to pursue my passions. I'd also like to acknowledge the National Institute for Aging for providing animals from their aged rodent colonies. And a big thanks to IPN and everyone involved in this conference for all their hard work and for allowing me to present my research. Thanks everyone for listening. If you have questions or would like to talk to me more, you can contact me via the email on your screen, and be sure to follow me on LinkedIn for updates to my work.